Hello, everybody. Thanks for joining us today for our webinar series on animals, climate change, and public health. Today, we have the second session, and the second session is on animals in research, and we focus on COVID-19 research. And just some housekeeping. We have the Q&A box that you can use to put your questions, and please put your questions in the box while they come to mind. And what we will do, we will have 4 short presentations by our experts, and then we will afterwards have a discussion and address as many of your questions as we can. So that's how we would do it. And please don't use the chat option because it's a little bit we are too many participants to answer and address all the questions otherwise. So why are we talking about COVID-19 research? As you have seen in the media, a lot of animal models are currently tested and used for COVID-19. We have, for example, seen that Syrian hamsters are used, ferrets, genetically modified mice. Mice are generally not susceptible for the new coronavirus, so they have to genetically modify these animals. We will also talk about that later, if this is useful or not. Also, pigs, dogs, cats are used, and non-human primates. There has been an outcry by animal researchers that there's a shortage of non-human primates at the moment. We will also talk about this in a little bit. And now I want to introduce our first speaker, and that is Dr. Aysha Akhtar. And she has a master's in public health, and she's the president and CEO of a newly founded center, the Center for Contemporary Sciences, which is catalyzing the world's transition away from unreliable animal experiments to innovative medical research and testing. And she's the author of the book, Our Symphony with Animals on Health, Empathy, and Our Shared Destinies, a book I can highly recommend. I loved reading this. So Aysha, the floor is yours.
Okay, thank you everyone. Thank you for joining us. So what I'm going to do is start the session with a sort of a broad overview of the scientific issues for animal testing. And since we have a very broad audience, it's for the scientists out there, this may seem a little bit more lay friendly than what you would expect, but I think it sets the foundation for the subsequent talks that we're going to have. So let me go ahead and start sharing my screen. So when we talk about animal experimentation, usually, you know, when we talk about the ethics of the issue, we tend to think about the ethics as in regards to the use of animals and how it affects animals. So what I'm going to propose is that what we really need to start doing, in addition to talking about the ethics of animal experimentation as it relates to the animal welfare benefit, is also to talk about ethics when how it pertains to human use. And so here's a photo of my dad, or my Abu, as we say in Pakistan. Now, my dad has suffered from diabetes for most of his life, and as a result, he frequently gets a severe type of nerve pain. And this type of pain is different from your average pain, and it requires a different type of treatment. And I have seen my father suffer again and again throughout the years from this pain, with quite honestly very little to offer him. And it has been an incredible source of frustration and sadness and grief for me to see him suffer through this pain without any reliable treatment for it. I've also seen, I worked for 10 years at the Food and Drug Administration. I've seen drugs come through the pipeline again and again that scientists in the healthcare world thinks is going to be great for a variety of diseases, only to see these drugs fail when we try them in humans. And most recently, I was Deputy Director of the Army's Traumatic Brain Injury Program. And I have to tell you, we have nothing really to treat traumatic brain injury in the soldiers. And that's been another source of frustration. all we can offer them is supportive care. So what I offer is that we need to reframe the ethics when we talk about this issue. To not only ask, is animal experimentation ethical for animals? Of course it is not, but also, is animal experimentation ethical for humans? And in order to answer that larger question, we need to ask ourselves, is it an effective use of our resources, and does it reliably inform human health? Great. Okay, so this is the picture of my dad, and these are the questions that I was discussing just earlier. So let me go ahead and move on to the next slide. This is what we call a pyramid of evidence, and in the biomedical research community, we have this hierarchy of evidence. And what basically that means is that at the bottom ring, the type of evidence you see at the bottom of the pyramid, it's considered the weakest form of evidence in the biomedical sciences. And as you move up the pyramid to the top, that is considered the strongest form of evidence in the biomedical sciences. And if you take a look at an all-encompassing look at the biomedical sciences and what's published in the medical literature, what you find is that in general, the evidence in support of using other animals, using other species to inform human health tends to be at the bottom of the pyramid. In other words, the evidence for the use of animals tends to be the type of evidence, it tends to be just expert opinion. So I know we've all heard this many times. We'll hear people say animal experimentation is the gold standard in research without really giving real evidence to really say why you have that opinion. And we'll see similar types of expert opinions that are proposed in support of the use of animals to inform human health. On the other hand, the evidence that's coming out that is largely in support of moving away from animal testing because it does not reliably inform human health tends to be at the top of the pyramid. That type of evidence tends to be what we call systematic reviews. And systematic reviews are when a researcher or a group of researchers take a look at the totality of data that's available to the public, the totality of information about a particular question, and they provide an exhaustive summary of the evidence available. So in other words, or a conclusion, is that the evidence, the so-called evidence that's largely in support of the continued use of animals is at the bottom of the pyramid. It is considered the weakest form of evidence. And in opposition, the evidence in support of moving away from animal testing is among the strongest forms of evidence. So let's take a look at some of the data is showing. If we look at basic research, and basic research is the largest use of animals in biomedical sciences. It is where most animals are used and actually where very little attention is drawn to. And this is where our tax dollars tend to go towards when we're talking about basic research. And this could be using animals in the lab to study basic biological mechanisms or to study basic mechanisms of a disease, for example. There's no applied benefit that comes out from this basic research. Well, in 2000, the Institute of Medicine here in the United States, which is considered a very prestigious medical body, took a look at basic research, and they took a look at what develops out of all of this basic research. And they concluded and published this conclusion that despite the great promise that comes out of basic research, it has led to very little tangible human benefit. That was in 2000, and there's no indication that that has improved in any sense. If we look at applied research, and here I'm talking about drug development, I'm sure many of you have seen this number here. About 15 years ago, the Food and Drug Administration had come out and stated that about 92% of the drugs that go through the drug development process fail in clinical trials. And that number ranges anywhere from 90 to 96%, depending on the disease category of interest. And that number has not changed over the past 15 years, despite many, many attempts to improve the use of animal testing for drug development. And if we look at why why there's such a high failure rate in drug development. So several explanations are brought up. One is that regulatory hurdles are potentially becoming higher and higher. The other is that clinical trials are very long and expensive, and those two possibilities are very much probably very accurate. But the third reason that's increasingly coming to light is the poor predictability of animal experimentation. And so what we are seeing is about 30% of the drugs fail in the clinical trial stage because of toxicity effects in humans that were not predicted by the preclinical tests. And that includes animal experiments as well as traditional in vitro tests. And about 70% of those drugs fail because they end up not being effective in humans. So let's look at some of the main reasons. I offer 3 main reasons why animal experiments often fail to reliably predict human outcomes. And the first is the uncontrollable influences of the laboratory environment and procedures. And what that means is that studies have shown that just about anything you can do to animals in the lab can affect the study results. So we're talking about the humidity level in the laboratory, the lighting in the laboratory, the caging procedures in the laboratories, the type of food animals are kept, where the animals are purchased from, what buyer, what seller they're purchased from, how the animals are handled, how often they are handled. You can go down the road. Just about anything you do to animals in the lab can influence the study outcomes in a way that we cannot predict and in a way that we cannot control. For example, one study looked at 2 different types of caging systems, and they were looking at genetically altered mice. These were mice that were genetically altered to display a cardiac defect, basically to display holes in the heart. And the study showed that the mice, whether the mice displayed this cardiac defect depended on the type of caging system they were in. And there were other studies that show similar results. A second reason why animal testing may fail to produce reliable results that we can apply to humans is that in animals and labs, we tend to be artificially inducing in these diseases. So for example, if we look at stroke is actually a relatively easy disease to understand. And in humans, stroke is mostly the result of a sudden disruption of blood flow to a certain part of the brain that starts causing cell damage and cell death. Well, in humans, stroke is usually the result of years and often decades of underlying physiological changes and the result of predisposing conditions like diabetes, obesity, hypertension, atherosclerosis, inflammation, and so on. Well, animals in labs tend not to get stroke naturally. So researchers will try to artificially induce the disease in these animals. So what that means is, in general, what they tend to do is either induce a blood clot into the brain to travel up to the brain to clog that blood vessel to create the symptoms of a stroke, or they will clamp down on a blood vessel. The problem with that kind of way of artificially inducing a disease is that you're not recreating all the underlying conditions that cause stroke in humans in the first place. So researchers have said now we need to create these underlying conditions. So in this photo, you have an image of an artificially induced obesity in a rat. This isn't natural. Rats don't tend to suffer from obesity in general, but this was artificially induced. The problem with artificially inducing the underlying conditions is the same problem as trying to artificially induce the main disease of interest. They are still artificially induced. So you're continuing going around in circles when you're trying to recreate a human disease in another animal. And then the third reason why animal experiments may fail to predict human results is no matter what you do, you cannot get around the fact that ultimately a mouse is a mouse, a dog is a dog, a monkey is a monkey, and a human is a human. And in fact, no human is the same as every other human. This is why in clinical trials and human trials, we need to have a variety of humans participate. So we have men and women, people of different ages, people of different ethnicities, and so on, because one human cannot necessarily predict what's going to happen in another. So if we look at some of the systematic reviews that have really taken a hard look at the reliability of animal experimentation, one that really comes to mind is this sort of groundbreaking study that was published in 2007 in the British Medical Journal, and it caused a whole lot of hoopla and controversy at the time. And what the researchers did here, this was a systematic review, and they looked at 5 different diseases, 5 different, very, very different diseases. And they looked at 5 different interventions for those diseases, 5 different drugs or other types of interventions. And then what they did is they gathered all the animal data on those different types of interventions, and then they gathered all the human data, and they compared the human data to the animal data. And what they found was that the animal data predicted the human data only 50% of the time. In other words, the animal data was no more reliable in telling us what we were going to find in humans than a toss of the coin. Now, this was the first of many systematic reviews that have come out. If we look at stroke, which I mentioned is a relatively easy disease to understand, we can really see how animal experimentation has failed to reliably lead us to a good therapy. There have been about 700 stroke treatments that have been tested in animals and have been found to be effective in animals. Out of those, 150 have moved on to human clinical trials, and not one of those has proven effective in humans. If we look at HIV vaccines, because we're going to be talking about vaccine research for COVID a little bit later, more than 85 potential vaccines have been tested in animals. Of those, 30 have moved on to human clinical trials. Not one has proven effective in humans. In fact, some of those vaccines have proven to increase the risk of HIV in humans. And just this past year, in 2020, in the spring, there was yet another HIV vaccine that failed. And this was a vaccine that was a result of at least 10 years just in organizing the clinical trials alone, and more than $100 million spent to develop this vaccine. And that failed again when tried in humans. One of the other problems with relying on an unreliable system, i.e. animal testing, is not only the failure rate that comes about, but also the potential for missed opportunities. So this is kind of a general outline of the drug development process. And I will say the last part of the outline here is incorrect, because what would really be more correct is we see 1 approved drug that comes out of 10 compounds in clinical trials, not 5 compounds. But nevertheless, the point I want to bring up is that let's say about 250 potential compounds, potential drugs or vaccines are tested at the preclinical stage. And this includes animal experiments and traditional in vitro studies. Out of those that are tested at this stage, maybe about 5 will move on to the human clinical trial stage. So that means about 245 potential compounds did not move on to human clinical trials. The question we need to ask is, if there's such a high failure rate with the compounds that do move to clinical trials, what's the flip side? In other words, how many missed opportunities were there? How many potential useful compounds did not proceed to clinical trials because of unreliable and misleading results in animals? How many drugs that should have moved on to clinical trials didn't, but would have been wonderful, would have worked tremendously in humans? This is a question that's really hard to answer, but there's no reason to think that there would not be many, many missed opportunities. So in summary, I'm sorry, here's the clinical trial. the pipeline here, for the drug development pipeline. So in summary, when we're talking about human harms, there are potentially three ways that humans can be harmed. The first is adverse events that occur in clinical trials or post-clinical trials in humans that were not predicted by the animal tests. The second is a significant wastage of time, money, and resources. And I'll just recall that HIV vaccine that failed, after more than $100 million was invested into that vaccine, and 10 years was invested just in the clinical trial phase itself. And then the third potential human harm are missed opportunities. How many drugs, potential useful vaccines, could have worked wonderfully in humans, but were discarded because of misleading results in animals? So what's the solution? I'm just not really going to go into great detail into these solutions, because some of our subsequent speakers are going to go more into more details, but there are some wonderful techniques that have been developed and continue to be developed that are based on human biology. So in essence, the solution to the problem is that biomedical research needs to start and end with human biology. We need to get back to understanding human biology, not to focus on the biology of a rat or a mouse or a dog or a cat or a monkey. We need to focus on human biology, and these are just some examples that will be discussed in more detail in the future, down the road in the seminar. And just a recap of what my organization is doing. As Kat mentioned, we have a new organization here in the United States. It's called the Center for Contemporary Sciences. We just launched in May, so we're a brand new organization, but we're a scientific organization whose aim is really to catalyze the transition to human-specific medical research. We know this is where we need to be going in medical research. It's a win-win situation for both humans and animals, and it is the future of biomedical research. And there are several ways we're going to be doing this, and you can actually kind of think about ignoring this next slide. But basically, there are three things that we're going to be focusing on. The first is to increase confidence in these new techniques. So there are a lot of new techniques already out there, and as I mentioned before, they continue to be improved and refined, but there are not a lot of people using these different tools. And part of the reason is there are many, or if not most, biomedical researchers have built their careers using animals. So in order to switch that, we need to help them gain confidence in using these new tools. So that means that we're going to be holding workshops that are going to help bring these tools into the hands of the researchers and give them that confidence. The second big focus of the Center for Contemporary Sciences is to create a workforce of new scientists that are, have been trained in using these new tools, basically to flood the field with scientists who are using these human-specific tools instead of using animals for biomedical research. And the third focus of the Center for Contemporary Sciences is really about money. Ultimately, the research is going to go where the money is. Ultimately, the progress is going to be made where the money is. And that means we are going to be working with investors to help match them with the entrepreneurs who are creating these new tools. We are going to be working with governmental agencies and charities to help direct them and help educate them and provide pathways for them to put more of their funding into these new tools. And of course, if that doesn't work, lastly, we'll be working on policy change to really kind of push our governmental agencies to be putting more and more of their money into these new testing methods and less of it into animal testing methods that have been proven again and again to be incredibly unreliable. So with that, I will go ahead and end. Thank you all very much, and I look forward to the discussion that will be following.
Thanks so much, Aysha. That was super interesting. Yeah, so our next speaker, I'm going to introduce her. So our next speaker, is Lindsay Marshall, PhD, and she is biomedical science advisor at the Humane Society of the United States and the Humane Society International. And Lindsay is the European advisor for the Biomed 21 collaboration. And if you haven't heard about this collaboration yet, it's really interesting to check it out, because they bring together scientists and institutions from across Europe, Asia, and the Americas, who share a vision for a new human-focused paradigm in healthcare. So without further ado, please go ahead, Lindsay, and start with your talk. Thanks.
Thank you very much, Kat. And it's really good to be here. It's good to be here virtually. I'd much rather be there physically. I'd much rather be able to see you all. And that's really because I'm going to start with an assumption. And my assumption is that you, our respected audience, are not giant mice. And I say that assumption because that's kind of what underpins biomedical research, is this idea that a mouse is a tiny person, or a human is a massive mouse. And so we can use them as surrogates of each other. And we've already heard from what Aysha says that that really doesn't work. So why do we carry on with that? It just doesn't make any sense that we are not mice. We are not giant mice. We're very, very different to mice. What I'd like to focus on for the next sort of 10 minutes or so is airways. My favorite organ is the lungs, and I'd like to talk a little bit about lungs. And we know how much COVID-19 affects the lungs. And if we look at mice and people, there are obviously similarities. We're both mammals, but if we look specifically at airways, there are huge differences that are incredibly significant when it comes to a disease like COVID-19. Now, you might already know that mice don't get asthma. Mice don't get chronic obstructive pulmonary disease, which is a smoking-related lung disease that people get. Even if you put a mouse in a box and fill it full of cigarette smoke for days and days and days on end, the mouse will not get the same lung disease that a person who smokes does. Mice don't get cystic fibrosis. This is a genetic disease that people have, and unfortunately, lung infections for people with cystic fibrosis often prove fatal. You can fill a mouse's lungs with bacteria if you've modified it to have cystic fibrosis, and it's not really bothered. It's not the same. And some of that is due to the differences in the airways between mice and human. So it's not just about size. Mouse lungs are incredibly different to human lungs. They have a different cellular composition in their airways. They have different proportions of different cells at different levels of their airways. They have a different architecture to their airways. So there are these huge, really significant differences that mean it's very difficult to look at what happens to a mouse and map it onto a person. And we see that, we call that a translational failure. And as Kat said in her introduction, mice are not naturally susceptible to COVID-19. So in order to get a mouse to even start to look like a human with COVID-19, you have to modify that mouse. You have to put a gene in it that is going to make it express something that COVID-19 would like to stick to be able to infect it. And by the time you've done that, really, how realistic is that going to be? And if we're looking at an infectious disease, then we've got to think about the immune response. We like immune responses. They're good for us. They help us out. They're very different for mice and people. A mouse lives in a different environment to people. They're small and they kind of scurry about on the floor. It's pretty dirty down there, certainly on my kitchen floor. So the mice have developed different defenses to people. We're a bit bigger, we're higher up, aren't we? We're not kind of in such filth as mice and other rodents are. So that immune response is different. So we've got the response to the infection is going to be specific to the species basically, and specific to the pathogen. And so we're not going to get that ability to work out what's going on in a person by looking at a mouse. So, okay, so people have moved closer to people. Researchers have moved closer to humans. So we look at non-human primate models because then, presumably reducing that genetic difference is going to improve the translational ability. Well, we've already heard that that doesn't really work. I think it's really sad that there is now a shortage of monkeys in the US because they're being used to test COVID treatments and vaccines and infections. And really, it doesn't really seem that that's the most efficient way to go. And not least because the non-human primates that are given COVID-19, they can be infected with it. So there's some susceptibility, but they don't develop the same severe lung disease that people do. So we can't look at that long COVID and we can't look at that severe COVID by using a non-human primate because they don't get the interstitial pneumonia. There are just too many of these differences. So this all feels very bleak. but it doesn't have to be very bleak. And what we can do are use the human relevant tools, these amazing technologies. So let's say 60 years ago, all we had was a mouse. That was our furry test tube and we couldn't do anything else. We don't really have that excuse anymore. And that kind of historical dependence on animal models is no justification for the continued use of animals now. And I'd like to just talk about two examples that have been used for COVID-19 research for respiratory disease, and that's the organoids and the organ chips. And if we start with organoids then, these are effectively miniature organs. So not a complete carbon copy of your lungs, obviously, that would be crazy, it would be beautiful, but it'd be, you know, impossible just now. But these are elements of your lungs. So you can take nasal cells, and in the lab, you can turn them into a nasal organoid. So you can create something that is like a miniature version of your nasal passages, basically. And with that, we can put COVID-19 virus on there to see whether or not it infects. And crucially, we can see where does it infect? What are the susceptible cell types? What are the regions of the human cells that that virus is attacking? And in the early days of this infection, we really needed to know that, because we needed to know what do we need to start thinking about for drug targets? How do we start to even begin to understand the pathogenesis of this pandemic without any information? So these organoids are really helpful for that. And we can build them from different areas of the airways as well. So we can do upper respiratory tract, so nasal and tracheal. We can do lower respiratory tract. And it's kind of important that we can look at these different regions. And the fact that we're using these different organoids from these kind of precise regions in the airways gives us a lot more information than infecting a mouse, because we can look specifically to see which of those different cell types, as we move down the airways, are susceptible, which ones are likely to be damaged, where do we need to think about protecting. If we're developing a drug, do we need it to stick up in the upper airways to stop the virus getting in, or do we need it to get all the way down to those lower airways to be able to protect us there? So that's one of the kind of advantages of the organoids, is that it allows us to get that regional specificity and that cell type specificity and that kind of responding specificity. So we can look to see which of our cells are producing alarm signals, which ones are not really bothered. What are those alarm signals? How can we exploit that to be able to treat this virus more effectively? So that's all I'm going to say about organoids. Then, because I'm more excited really about the organ chips, and the organ chips are a more dynamic system. So we're getting a step closer to recapitulating the human lungs then. And the lung chip won the design award in 2010 in London, and it is the most beautiful thing. And these chips they're chips, they're silicon chips, and they have channels in them, etched into them, and you can put your cells in the channels. And what we can do with organ chips is put different cells together to see how do they like to talk to each other. And the other thing that we can do is make these dynamic. So for a lung chip, what's incredibly important is that it breathes, because that's what your lungs do. That's what they should be doing. They should be moving as you inspire and expire. And that stretch is really important functionally. So it's great that we can start to recreate that now in our organ chip. We can stretch the cells and make them think that they're breathing. And underneath those lung cells, we can put the endothelial cells. So you're probably already aware that gas exchange is when the air comes into your lungs and it goes straight into your blood, and that's where we want it, so you can get it around the body. So there's very close contact between the lung epithelium and the endothelial cells. And that's what a lung chip can recreate, that close contact, but also the movement, not just of the breathing, but of blood flow as well. So we can send our kind of blood substitute through that endothelial channel, and not just our blood substitute, but we can put immune cells in there as well. And we can send them through the channel, and we can put virus in our air channel, which is where our lung cells are, see whether or not we can get our virus to stick and infect. And our lung epithelial cells will be pretty cross at that and send out signals. And we can look to see how do those immune cells respond to those signals in real time. We can image them to see what are they doing, which ones are interested enough to stop and go and have a look and try and do something and move out. And it's kind of experiments like that that give us the clarity, that tell us that the immune response in COVID-19 is different, the cellular response is different. So we're not looking at our kind of typical immune response. We're not looking at our, for those of you who like immunology, our polarized Th1 versus Th2, but it looks like we've got a kind of mixture of those responses. And I think we can only do that by teasing out the different cell types and then kind of putting them all back together again. So we take a reductionist approach and, you know, pull it all apart, and then we use those different segments to build it all back up again. But it's all human. So it all makes perfect sense, and it should all translate. The other thing we can do with our beautiful dynamic lung chips is we can sample what those cells are making. And again, we can do this at different time points. Really, really difficult, really stressful and challenging to do that in a mouse, to just keep taking samples from its airways and keep taking samples from the bloodstream at, every minute or so. But the lung chip doesn't care. You can do that to the lung chip. You could automatically do that to the lung chip. So you can look at the dynamic kinetic response of cytokine release by your epithelial cells, by your immune cells, by your endothelial cells, by all of those different players that are working together to be able to defend us effectively, so we can see what's gone wrong. And we can also think about, we've got that air channel and the blood channel. We can think about delivering virus to the air channel, but we can also use this to deliver an inhaled drug to the air channel to see, is that the best way to deliver these drugs? If we imagine somebody breathing in the drug, we can do that in our lung chip and see, okay, how much do we need? Where does it get to? What does it do? Or we can mimic somebody taking a drug orally, because we can put the drug in the endothelial channel, in the blood side of that lung chip. And then we can see, okay, as it flows across there, what happens then? What does it do? Is that more effective? So we can really get an absolute ton of information from these chips. And that information is all relevant to us and to that specific infection. We can put multiple different cell types in there. So we can look at cellular communication. We've got a kind of an ability to do that sort of automatic sampling. We've got the ability to look at the kinetics of these responses, and none of this is really possible in the mice. So it's really time to turn our backs on the mice and the animals and look at these tools that can really answer the questions that we need to ask. And I guess there's an element of expense in this as well, isn't there? And expense of animals' lives, but also expense as in, it's very costly to create new drugs. We've already seen that. A lung chip, as small as it is much, much smaller than a mouse even. So if you're creating a new experimental drug, you might only have a tiny microgram of it, but you could test that in a lung chip because you have enough of it to be able to look to see whether or not you can get any impact on several lung chips. But you wouldn't be able to do it on enough mouses to be able to get those data. So they become more economical in that respect if we're looking for new drugs. They become as well an incredibly useful tool for drug repurposing. We've heard a lot about this in COVID-19. This is a new pandemic. We have no idea what to do or how we're even going to start to address it. And this idea of drug repurposing is very popular. It's a very sensible way to go. And what this means is that a drug that has been previously approved by the regulators, and therefore we know a lot about its pharmacokinetics, and we know a lot about its safety profile. We know about its efficacy for a particular disease, but obviously not COVID-19, but it's kind of passed all of its tests. Well, the lung chips and the organoids, actually, these human relevant tools offer us a really fast and effective screening platform to see whether or not any of these drugs will be able to stop the virus replicating or prevent the damage or reduce those cytokine storms, or actually just, give us that protection against COVID-19. So what do we do then? What do we need? I think Aysha's already touched on that. And I agree with everything that she said about how do we try and improve the uptake of these methods. I think we need to fund these more effectively in terms of prioritizing funding for these human relevant tools. So we're trying to promote them and talk about them and say how fantastic they are. We need to train people how to use them, but we need to give them the capacity to be able to get money to be able to do it. And at the moment, the funding is pitiful compared to your classical kind of animal dependent biomedical research. So in Europe, it's less than 0.1% of the animal funding will go towards non-animal methods. So we need to kind of equalize that sometime, tip that balance somehow, so that people that want to use the non-animal methodologies are supported to do so. And we also need to improve confidence in our scientists to be able to use them, but in the regulators as well. So the people that have to make these really difficult decisions about whether a drug is safe or not, and are not quite sure whether an organ chip is going to give them what they need, then we need to be able to give them the confidence that they can say, yes, that it'll be okay. And I'm hoping that that might be a beautiful segue to my colleague Elizabeth Baker, because I am going to stop right there. So thank you very much for listening.
Thank you very much, Lindsay. That was super interesting, and we will go into more detail in the discussion. There are already some questions in the Q&A box, also from the first talk or for the first talk. So our next speaker is Elizabeth Baker, and Elizabeth is an attorney, and she is the Pharmaceutical Policy Program Director of the Physicians Committee for Responsible Medicine. And PCRM is a nationwide organization of physicians and laypersons that promote preventive medicine, especially through good nutrition. And they also promote higher ethical standards in research. And Ms. Baker works to modernize drug development to save human and animal lives by shifting non-clinical research from the use of animals to modern technologies that are more relevant to human biology. So I hand it over to you, Elizabeth.
Okay, thanks so much. I do, before I get started, want to just take a few moments to give out my thank yous. First, to the participants for spending your Friday with us. It's really great to see that we have close to 200 people who have decided to spend a few hours learning about these issues and hopefully engaging with us in the Q&A. Also, thanks to the other speakers, Aysha and Lindsay, you guys did a great job. I'm sure, Thomas, you'll have some really interesting things to say as well. And then to Kathrin, Andre, Charlotte, Ava, and the other organizers, a big thanks. I know going virtual has been tough for all of us, and it seems like hosting these virtual events include just so many minor things that make it maybe even more difficult than in person. So really appreciate it. Thank you. So I have been asked to talk about why we still rely on animals, and really kind of taking the regulatory framework into account. And there really are so many reasons that, unfortunately, these tests continue and the research continue. And I think Thomas might touch on some of these in his presentation next, but animal research and testing, it's built into systems, it's ingrained in systems, it's in our grants, it's in our journal review. There's so much organizational inertia that keep things going. You have scientists who have been trained and who understand how to use these methods, but don't have that experience with the newer approaches. You have different businesses and industries that are really conservative when it comes to changing. And then finally, you have a regulatory framework that demands it. And this is the case for drugs, it's the case for vaccines, medical devices. But today I'm just going to focus on drug development and take a deep dive into the regulatory framework. And I think this is really important because even if we have the best science and we have some really great methods, hopefully we'll hear more from Thomas. Lindsay did a great overview of some of the approaches that are available to us for studying COVID-19. But even if we have these great methods that are predictive for humans, that save on costs, that streamline drug development and are more efficient, if our regulatory framework and our written policies continue to demand these methods, they're going to continue to be used. And so for pharmaceutical development, all medications are tested extensively in animals. That's the case now, and it's been the case for about the last 60 years. For pharmaceutical development, the animals used most frequently are monkeys, which may be why we were hearing about these shortages, mice, rats, and dogs. And Aysha showed a nice overview of the drug development pipeline. Essentially, the very simplified version is you have your compound that starts in the lab, it goes into the non-clinical studies, and traditionally those have largely been the animal experiments. If those go well, it moves into different phases of clinical trials before it would ultimately be approved and marketed for sale in the US. And it is our United States Food and Drug Administration that is doing the regulation and overseeing this process. There are two main ways that I want to talk about that the FDA communicates requirements or expectations with drug sponsors for the development of new drugs. And the first one, well, I guess I'll just tell you what they are: regulations and guidance documents. Regulations are rules that an agency puts forth in order to help carry out their legislative mandate. They are binding, and they are to be followed by drug sponsors as they are developing a drug. Now, we have been working on this regulatory issue for a number of years now, and we are making some progress. And I think as a result of the work that we have been doing, we saw in a recent publication by the Food and Drug Administration that they meant to address this issue. And they say that the FDA regulations are inherently flexible to accept in vitro or in vivo data. And it was great to see that in print, because I think it reflects the intention of the agency for the regulations to say this. But unfortunately, it's not true of all regulations. It certainly is true of, there are many FDA regulations that do allow flexibility, but there are many that don't. And the ones that don't need to be changed. And I'm going to just share my screen here quickly to show you an example of what I'm talking about with the regulations. Okay, so we'll start with 21 Code of Federal Regulations, Section 312. This is a regulation that is on the books. It's in our Code of Regulations. It's for drug sponsors to follow as they are developing a drug. And it says that industry should provide a summary of the pharmacological and toxicological effects of the drug in animals, and to the extent known in humans. And so you can see the face of the regulation right there is asking for animal data. And so a company who is following the regulations is going to provide animal data. What we have proposed is that we switch out that word animal for non-clinical approaches. And there is actually already a definition for non-clinical in the Code of Federal Regulations. Currently, it covers in vitro and in vivo or animal studies. And we have asked the agency to amend that definition to also include computational approaches. So non-clinical would then, in the Code of Federal Regulations, cover the animal experiments, in vitro, and computational approaches. And then we've asked the agency to actually use the word non-clinical consistently throughout the regulations, because right now there are some references to non-clinical, some to animals and in vitro. So if we just stick with that word non-clinical, then it covers everything, and it opens the door for these companies to confidently use and submit approaches that are based on human biology and that don't use the animal, don't rely on animal data when they decide that these approaches are safe and are useful and they want to use them in drug development. So now we have been working on this for a number of years, as I mentioned, and we have had some recent success in this. We worked with Congress, and Congress ended up passing some appropriations language. So this is language that is tied to FDA funding, and it directs the agency to update these regulations and to report back by September of 2021. Now, of course, September 2021 is a long ways away, and so we are continuing the work. We're continuing the work otherwise as well. Now, the next thing I want to talk about is guidance documents, and I'll share an example of FDA guidance. So guidance documents are written policies that the agency puts forth. They're not binding, unlike regulations, but they do reflect the agency's thinking on a topic. And so drug sponsors look to guidance to help them as they are designing their package that they will submit to the agency that includes the different studies. Many guidance documents favor animal tests. Either there would be an enumerated animal test, or it would be more general, like saying two species, a rodent and a non-rodent. But increasingly, we are seeing that FDA is including language in guidance documents that really is intending to provide flexibility to use non-animal methods. but it's, that flexibility has not quite been made useful yet. And I'll show you just an example here in FDA guidance. So the guidance document that I'm referring to is the ICH M3R2. And this is a guidance document. ICH is the International Council on Harmonization of Pharmaceutical Testing Requirements. It's the FDA, European counterpart, Japanese counterpart, and others who have come together to outline, to try to harmonize testing requirements so that a company wouldn't have to do a certain set of tests for approval in the US and do other things for approval in Europe. And so this, this language is included in that document. It says that although not discussed in this guidance, consideration should be given to the use of new in vitro alternative methods for safety evaluation. These methods, if validated and accepted by all ICH regulatory authorities, can be used to replace current standard methods. And so it's great that that language is there. It really reflects the agency's intent to provide flexibility in this document, even if I think it's a very high bar what they're asking for here, but at least that intent is there. But here's the issue. So that is the ICH guidance as it was written by ICH. When the Food and Drug Administration adopted that guidance, they include their own little box at the top that says you can use an alternative approach if it satisfies the requirements of the applicable statutes and regulations. So we know that the regulations are requiring animal data. And so by including this language on the guidance, it essentially nullifies the intention to provide flexibility, or the flexibility, not the intention. And so I think this is something newer that we've begun working on, and I think that we're going to really start seeing some changes at FDA because the intention is there. It's more of working it out on the policy side, I would say. I'm going to stop sharing my screen now. I do want to finish up just talking a little bit about COVID-specific guidance. So the guidance that and the regulations I talked about, they apply to all drug development, and that does include COVID therapies, but other therapies as well. Now for COVID, the agency did put out a guidance document to help streamline communications between drug sponsors and the Food and Drug Administration in the pre-IND meetings. So in the meetings where they're planning what studies they're going to do in order to move on to clinical trials. And this, unfortunately, this guidance document does call for standard toxicity and general toxicity studies in rodents and in non-rodents. And so we are seeing that for all COVID therapies, that I think that's going to continue as long as that guidance is on the books. We have provided some input to the agency on this. We are in discussions, but I think it just reflects a larger issue. And that is that we need our regulatory authorities to commit to reduction and replacement. You may have heard that the EPA did this last year, and it really has helped to prioritize this within EPA, where it's kind of redirected the scientific community. It is redirecting funding. Programs are being established to make this happen. And so I'm hoping that the next agency we'll see make a commitment like this will be the Food and Drug Administration, because as I said, I think the intention is there. There are some programs that are being established, but some of these main issues have to be addressed. And certainly a commitment like that will free up resources to address these things. I think I should probably wrap it up in the interest of time, but I would like to close by saying, you know, I know it's hard to think about these issues. It certainly can be hard to work on these issues when you know about all the suffering that is involved for the animals. And then of course, feeling like, as Aysha said, we could be doing better for human patients who need safe and effective medications. But I really have seen a shift in recent years, and things are moving in the direction of human biology-based approaches, both in research and in product testing. And I think it's up to us, my colleagues on this call, to continue keeping it a priority and to continue engaging with the public and finding ways to get the public involved to show that this is what we want, and we're not going to stop until we get there. Thank you.
Thank you very much, Elizabeth, and thank you for giving us your hopeful outlook on the matter. And yes, that's one of the reasons we included this session in our webinar series, is we want to inform the general public today about the issues with using animals for human health research. And so, yeah, thank you for your work on that. And so now I come to introduce our final speaker. Our final presenter is my boss, Dr. Thomas Hartung, and he's the Dorenkamp-Simpson Chair for the Evidence-Based Toxicology with a joint appointment for Molecular Microbiology and Immunology at Johns Hopkins Bloomberg School of Public Health. And he also is a professor for pharmacology and toxicology at the University of Constance in Germany, where we have our sister center, CAT Europe. And Dr. Hartung is the director of both Centers for Alternatives to Animal Testing at both universities. And so I hand it over to you, Thomas, to start your presentation.
Thank you very much, Kat, and thanks for the previous speakers to set the scene about animal testing and drug development. You can hear me well? Looks good. Okay. Okay, then let me share my screen and get into it. I will focus today very much on the aspect of COVID-19, because I think that's an interesting game changer at this moment. So the question is COVID-19 research with or without animals. And you see me here wearing a mask, as most of us do. We actually printed these masks, a little bit smaller logo, but it says saving more than animals. And this is an important message for me. I'm a physician in a leading school of public health, and our fight for replacing animal testing by modern technologies is in the best interest not only of the animals, but also of patients and consumers. That's my strong belief. So what we are observing at the moment is what I like to call a scientific gold rush at the Klondike. Everybody who can just spell COVID-19 writes now a paper or a grant application, because all expectation is that scientific money will pour down on us if you're only in this area. The problem is that most people are actually quite ill-equipped. They have never worked on viruses and similar. And very few will get rich in this exercise, as at the time at Klondike. These are mostly the companies who provide the reagents and the tools. It is very rarely the researcher which just entered the field. I checked a month ago, and we had already 55,000 scientific articles since December on COVID-19 and the SARS-CoV-2 virus. 55,000 PubMed listed articles. So this does not include all the non-PubMed listed articles, and it is calculated about 4 times more medical literature is published. And it does not include all of the self-published archived articles, which we are also seeing as a rather new phenomenon. So this is enormous. And I think we will need some systematic review technologies, quality assurance and others to really dig into this material, but it also shows you how difficult it is to somehow hinder the scientific productivity which is taking place here. But it's without question a success story. If you imagine the world was made aware of this on the 31st of December last year. That's when China informed the WHO. And it was only 10 days later that we had the genome revealed. It was 17 days later that we had the first PCR diagnostic outside of China, which was then adopted by the EU. And on the 1st of May, already the first therapy which had shown benefit in clinical trials was receiving an emergency registration. So it's an enormous thing. If you just look into the number of clinical studies, this is unheard of. There were more than 20,000 studies launched according to the Cochrane Study Register on COVID-19, with more than 3,000 of them being interventional studies, so trying to find therapies. Sure, a lot of this is repurposing existing drugs, trying out what we have, but a lot of them is new stuff. As the number of vaccines, for example, shows, we have more than 160 vaccines which are under investigation. There's a handful already in phase 3 trials. Already in April, 4 months later, we had 115 vaccines under trial, according to the paper. A very important aspect of this is that we have to consider the economic aspects of what we are trying to achieve. We have to see that developing a drug or vaccine is primarily an economic aspect. And we have summarized some of our thinking here in 2018. What are the real numbers? What you can see here is a steady increase in the costs of developing a drug and a pretty stable timeline for this. Drug development is now at about $2.9 billion for one drug making it to the market. And you see here, the costs are split almost equally between preclinical and clinical work. And it takes on average 12 years to develop such a drug. Vaccines are no different. Average time is 10.7 years, but it varies from 8 to 18 years. The costs are a little bit lower. It's only about 1 billion according to the data I found, but it is a very costly exercise. The toxicology is interestingly an expensive part of product development, but interestingly, this has shifted much more than most people are assuming from in vivo toxicology to an in vitro approach. The pharmaceutical industry and others are using broadly cell culture methodologies. As you can see, there's about 4.2 billion spent per year for in vivo toxicology and 14.4 for in vitro toxicology, which shows that these methods, which we are advocating, are actually being used. Whether they're used enough, we can discuss, but I think there's no hesitance to start applying them, and this is important. You've heard from the other speakers that these are exercises which often fail. Roughly 95% of drugs which enter into clinical human trials fail. There's actually the success rate of new drugs is even lower because many of them are just Me Too developments, which means a company is trying to launch the same substance another has already shown, or one which is only slightly modified, and they want to show that it is similar efficacy, efficient, and bring it to the market. Another aspect which I find quite remarkable is the central role of the FDA in setting the games of the rule. And the reason is an economical one. Here in the US, we represent 4.25% of the world population, but we consume 48% of all drugs, and we consume 64% of all drugs under patent. So it's very clear, the companies are only interested, does the FDA accept the methodology or not? And for this reason, FDA's very strong involvement and commitment to the new microphysiological system, and others you have heard about already, is of critical importance. If you want to know a little bit more about the role of alternative methods as in contributing to drug and vaccine development, this is an article which is open online available, which we wrote in Archives of Toxicology a bit earlier this year, and where we discussed the need for models for the various aspects of the drug development cycle. For drug repurposing, so using drugs which have been used in the past for other things, for target discovery, for drug efficacy testing, for vaccine development, for combination therapies, for drug safety, and for their quality control. And this is something very important to keep in mind. The quality control, especially of vaccines, is one of the most animal-intense uses which we have. Per year, we are testing, for example, about 500 different batches of human influenza vaccine in animals, which is a major consumer of animals. Vaccine quality control for batch release consumes in Europe more animals than toxicology. This is something where we had a lot of progress in the past, but where we also still have a lot of room for improvement. A very important aspect is how to test if you're using animals. And there's actually already a crisis identified that we are running low on COVID-19 vaccine tests for in monkeys, because rhesus macaques became a rare commodity. Among others, because China, which is responsible for 50% of the supply in the US, stopped exporting rhesus macaques to the US. And for this reason, the supply here is actually pretty short. I discussed the aspect of why rhesus monkeys are used in the India Times a little bit earlier. If you're interested in my views on this, I'm happy to send the article, but you can also probably Google it. And what I think are a few important take-home message from this interview. We should first of all be aware of the extreme diversity of the immune system. The immune system is under evolutionary pressure every single day, because we are fighting infections which you never will suffer from, because we have the immune system. And it is for this extreme evolutionary pressure so diverse, from one individual to the other. If you take two inbred mouse strains, they will have a completely different immune response typically. The problem with these rhesus macaques is we have extremely small group sizes. And this is even enhanced by the fact that these have to be done now under biosafety. So in the protective gear for experimenters, so we will have very, very few animals only in each group. Typically not enough to make any predictive measurement. And species differences are enormous. COVID-19, to the best of my knowledge, only leads to a relatively mild respiratory infection in these macaques. A lot of what we see in our patients is not the case here. And we know that from the sad example of Tegenero, an antibody which was tested a decade ago in monkeys at 500 times higher doses, and the first 6 people which were treated were in intense care 6 hours later. So a monkey is not a little human, that's for sure. In humans, we know we first of all have a respiratory disease, but we have learned over the last 9 months that this is actually affecting a broad variety of organs: the kidneys, the liver, the gut, the heart, the brain, the blood system, where it's leading to clotting and strokes and others. Rodents do not at all show this infection. You can render them susceptible by transfecting them with the human receptor, the ACE2 receptor, but then you don't know, do you put this receptor into just some lung cells? Do you put it into all cells of the body? It is definitely not the natural course. The rhesus macaque, but also hamsters and ferrets, show a relatively mild infection to the best of my knowledge. It's a respiratory disease. They do not show, as far as I know, any affection of other organ systems, as we have seen in our patients. And I think it is also rather unlikely that this becomes really a major systemic infection if the respiratory part is relatively mild. Interestingly, human cells, especially in these microphysiological systems, these organs on chip, these organoids, they check most of the boxes. For all of these organs, we have publications in the meantime which use these type of systems. And this is an important message, I think, for everyone. Tissue culture has evolved in the last decade in a way we could not imagine. These are no longer pan-fried eggs in a pan, in a petri dish in this case, cooked sunny side up with a big nucleus and hardly any cell-cell contact. We can reproduce organs. And a lot of this was fueled just by the development of induced pluripotent stem cells in 2006. So not that long ago. And only since then, different organ systems are actually mushrooming, and we put them into microfluidics and we put humans on a chip. On the right, you see two stakeholder workshops, which we did carry out. Each of them more than 30 participants from both sides of the Atlantic and some Japanese. We just published the second one and discuss where is this technology and what are the challenges ahead. And I invite you to read them in our journal Altex, which is free of charge, openly available. To show you how big the expectations for this technology is, I would like to show you just one quote from Francis Collins, the director of the NIH, who wrote in 2016, showing one of these biochips for lung actually, said, "I predict that 10 years from now, safety testing for newly developed drugs, as well as assessment of the potential toxicity of numerous environmental exposures will be largely carried out using human biochips. It will mostly replace animal testing for drug toxicity and environmental sensing, giving results that are more accurate at lower cost and with higher throughput." And as a consequence, the NIH, the FDA, and others have invested more than $300 million into the development of these microphysiological systems. I think this is something which is really remarkable. If you're interested, we are in preparation of a World Summit on Microphysiological Systems. We hope to have in December 2021, though this is under discussion. You see the co-hosts of this event, Susie Fitzpatrick of FDA and Don Ingber from the Harvard Wyss Institute, one of the iconic protagonists of microphysiological systems, will be co-hosting this. And we have in the meantime some 36 organizations, including beside here FDA, also the European Food Safety Authority and European Medical Agency and others. So please stay tuned for this event, which will take place either in December 2021 or shortly after. To give you a real life example, I would like to refer you to our own microphysiological system, to our own organoid, which is the brain sphere model. This brain sphere model was not the first mini brain. When we announced it, we were only the third or fourth group, depending on how you count. And, but we were the first to mass produce them, thousands of them every week in exact shape and composition. And this was very important for toxicological and pharmacological applications. These are electrophysiologically active, they show myelination, a lot of aspects of the brain, but they're also very simple in their setup. When it became clear early on that COVID-19 is linked to a lot of neurological manifestations. Already the first studies from Wuhan in February said 36% of the patients are showing neurological manifestations. And some newer studies from Europe suggest 50 to 60%. And I even saw one which suggested more than 80% neurological symptoms. So these studies prompted us to ask, is this possibly also due to some infection of the brain with the coronavirus? And indeed, we were able to show as the first group worldwide in a peer-reviewed paper that SARS-CoV-2 is infecting human brain cells. This is a neuronal cell, probably. It's very difficult to say from the shape, but from some other experiments, we are believing it is mainly a type of neurons which can be infected. And this was covered very quickly by a lot of press. You see here the Financial Times reporting on our results and others, which we made in May, June of this year. And this has a lot of important consequences. Here you see the core team which was working on this, and the paper published in Altex end of June, finally, because it might be an indication of derailed neurodevelopment, possibly due to virus infections. And there is more and more evidence increasing that the virus can pass the placenta, that the virus can infect the embryo, and possibly can also be found in the brain. There's studies showing that the virus might be persistent actually as an infection in the brain, which has a lot of consequences, possibly fueling Alzheimer, possibly being a lifelong risk for those which have even had only minor infections as a consequence of a virus infection you cannot get rid of. In our studies, we showed the presence of the critical receptor in some brain cells. We showed that some of these cells are infected, a very small proportion, and we don't know how much this will produce pathology. And we showed the replication not only by these beautiful pictures, but also by PCR technology. So let me close at this point. I think the message I would like to give you is these alternative methods are enabling technologies. As I tried to show you, we would not have been able to show that human brain cells can be infected with this virus with any of the animal models which is around. I believe we can ask a lot of things which we cannot do easily under biosafety levels with using these technologies. And I believe these technologies are now a door opener for a faster development of drugs, moving fast into humans, producing safety data quickly. But we will also ask at some point, if we can use them here, why can't we use them for other type of drugs? And perhaps COVID-19 also becomes a door opener for the new approach methodologies. Thanks a lot.
Thanks a lot, Thomas. That was very interesting, all 4 presentations. Thanks a lot. I would now welcome everybody from our experts to join for the discussion. Please switch on your cameras and unmute yourself. We have several questions, and maybe you also have questions for your colleague, one of you in the panel. I would say we could start with some transparency, if this is something that everybody is interested in, because obviously it's a big problem that there's a lack of transparency in animal research, but also we can also say that for clinical trials, for example, when is a clinical trial authorized, you know, what kind of data was available before that this decision is based on. Also, we could talk about the need for pre-registration. Those are some things that came to my mind during this. I just give the floor to you now.
Sure. You know, if that's what you're doing, I also wanted to address, if that's okay, Kat, to address some of the questions that.
Yes, we will do that afterwards. This is just, we will definitely come to some of the questions of our audience, but just to give a little bit more information. One thing that came up during your presentation, Aysha, was that there was a question about environmental enrichment. And how environmental enrichment of cages would, it sounded like, negatively influence the data you obtain. However, the opposite is the case. The stress of animals when they are not housed properly, that's definitely another issue. But I think what is important to stress here is that we want this research to be translatable to humans, which is why it's just still not good enough to give environmental enrichment or not. It is just not the question. So maybe to start out with that, we can focus a little bit on what the difference, to understand the difference between internal and external validity, because I think many of our listeners might not know about this, these differences. And also maybe some of you or all of you want to speak on the matter that, of course, the animal experimentation community is trying to improve animal experiments because we have this reproducibility and also translatability crisis. My personal observation is, and I'm wondering if you share this, is that there's a lot of focus on the internal validity. That means that you try to avoid bias in planning, conducting, analyzing your data, and then also that you avoid bias when you report your data. But I don't think this is far-reaching enough, because we need to also look at the external validity. So I would like to invite you, maybe Aysha wants to start to address this topic and what you also think about environmental enrichment and so on.
Sure. So there you know, when we think about, so yes, there's been a lot of discussion about how to improve the so-called animal models and how they're tested so that one animal study will predict another animal study to basically to recreate the results, to replicate the results again and again. And that's been one of the problems that's been brought up. I feel that that's really taken away from the real question, though. It's not about how to get one monkey study to predict another monkey study, but how do we get these animal studies, or how to get studies that are going to predict human studies. And so when I go back to that, you know, that one systematic review that I mentioned that was published in 2007 in the medical journal, and I think this leads also to one of the questions I saw. What it was really showcase how poorly animal results are going to predict what we're going to see in humans. So only 50% of the time did the animal results tell us, only 50% of the time in that study did the animal results match the human results. So that's an incredibly poor prediction. I mean, I would say that's not, there's a lack of predictive ability in animals to start to predict what's happening in humans. When we, you know, and so when we talk about even things like enrichment and all these other things that we can be doing to animals, you know, I forgot what I was saying, but one of the studies, going back to how enrichment can affect the animal's physiology, there was one study that actually did look at enrichment in monkeys in their caging systems. And it found that a certain type of enrichment, if you increase enrichment in monkeys, that changed their neurophysiology in comparison to the monkeys that did not get that enrichment. So even basic tweaking of their living environment, and this was a very minimal tweaking of the living environment between one group of monkeys and another, can cause differences in just their biology, let alone what those study results would be. I think, you know, we can sit here and tweak around with animals. The problems with using animals fundamentally gets down to the fact that we don't know everything about these animals. We don't know everything about human biology. We know so little, quite honestly, in comparison to what we don't know. And it's the same thing with every other species. We don't know. So it's all the things we don't know about what's unique to each individual species, to each species, that is ultimately causes the problems, ultimately leads to the failure in their ability to predict what we're going to find in humans. How can you improve on a system when you don't even know what that system is? It's like, you know, you can take a mathematical model and you can continue to improve it because it's a fixed system. But with living beings, you're talking about systems we're talking about beings that are our physiology, our biology, our behavior, fundamentally related to everything in our lives, to who we've interacted with in our lives, how we interacted them, to our environments, to different genetic makeup and expression and so on. We don't even know a full rat's biology. How can we possibly expect to try to apply a human disease in a rat biology if we don't even understand what the rat biology is in the first place? So I'll stop there and I'll let other folks continue on.
I agree, Aysha, with what you said, of course. And I think the point that you make about tweaking is a really important one. And I think for me, that links to what Thomas was saying about evolution of models. And it seems to me that the animal is the animal. You can tweak it as much as you like, but it's not going to change. It's got that mouse biology in it. And we don't really care about mouses. We've been able to cure them of cancer for years. So actually, let's think more closely about the evolution of those tissue culture models. I think that slide that Thomas showed was fantastic. You've got almost this exponential rise in the ability of those models to model the human system. And it's not just a single model now. We can start to bolt things together to start to look at organ systems talking to each other. And that to me seems to be a much better use of our kind of time and money and fabulous brains that these research scientists have, rather than tweaking a mouse, putting a different gene in it, trying to humanize it. And you've still got that underlying mouse biology. So I just like to advocate for these projects that are looking at humans on chips and patients on chips. And we're not quite there yet. But if we put more effort into that, rather than tweaking a mouse or a monkey, I feel like we'll get there. And we're going in the right direction with that. The mouse and the monkey, we're over here somewhere. but the human, we're on the right track for the human. So I think that's where we need to put our resources. And I think it comes down to kind of the things that we've all touched on today, is education and funding.
Yeah, I mean, if you see, I always say my mantra, we are not 70 kilogram rats. Yeah? And this is, I think, really giving you the central message. We are interested in something different, but we should be careful. We're also not 70 kilogram cell culture. These are all models. And I think we have to understand that, especially in the context of drug and vaccine development, where you're in the end betting on substances. You're spending hundreds of millions in order to either make a multi-billion-dollar revenue or have it all lost. And in this process, I understand that companies are using all information sources to make their decisions and trying to put the money on the right thing. The problem is they overestimate very often the value of these models, especially in toxicology. You know, in pharmacology, we are used that each and every test gives you a tiny little bit in your information making. You would never say there's one animal test which gives me all. In toxicology, this is different. We have a certain set of tests, and each test gives us an answer for whatever we want to know, we believe. And this is not really functioning. The problem is, however, also here that the companies don't dare to move away from this because they might make an entire registration not acceptable by deviating from what has been done. And if you see that one day delay in a drug being accepted by the FDA or so is on average $1 million lost. Yeah? One day, $1 million. Do you want to be the researcher in the company who said, yeah, I said we don't need this animal test, we do it in a cell culture instead? Do you want to be the researcher who said we don't need this test when the company has to defend their substance in court? And the court says, why didn't you do the animal test, which was always helping us to be safe for the last 100 years? These are economical questions we have to conquer. And the most important part of this is to create doubt, make clear to the people in a most systematic way that the animal test is not giving you all of the answers.
Okay, thanks a lot. That was were interesting answers. The other question we got from one of our attendees is, are there chips yet available which test interhuman varieties? Because we know that, you know, it's not only the interspecies differences we have, but also between humans. And is there already something on the way?
Yes, and first of all, the big advancement is with the induced pluripotent stem cells that you can produce them from different genetic backgrounds. You can have them from patients which have shown a certain disease. You have genetical backgrounds which allow certain diseases. And it is still costly to produce them on various stem cell lines, but it is the start into an individualized, personalized toxicology and pharmacology. There's no other technology which would allow you to do this, at least not human relevant. Yeah.
There's also, to add to that as well, there's also the National Center for Advancing Translational Sciences have just announced their recent award program. They are looking at clinical trials on a chip. So they're looking to use patient cells, develop the chip models of whatever organ it is that they need for their particular patient for that condition, and then use that for drug screening. So I'm sure you've all heard of personalized medicine. So this is kind of a fast route to look at personalized medicine. So to take, for example, a patient's tumor, grow it on a chip in the lab, in an organoid system, whatever, put those different drugs or drug combinations across that and see which one works for that specific patient. So there's a huge power in being able to develop these systems for individual people, individual patients.
And I think the FDA and the NIH definitely understand, as well as drug companies, that we need more diversity in drug development. There are quite a bit of efforts going on for clinical trials. And so we have just started pushing for diversity in non-clinical studies as well. And it would be supporting, like what Thomas is talking about, with the induced pluripotent stem cells from a variety of backgrounds.
The difficulty is that it takes you about half a year to make an induced pluripotent stem cell from a skin donation or whatever you use. from a human donor, a patient. And then it takes you another half a year, I would guess, for most models to adapt them to producing an organ or a disease model. So for decision taking on a patient, it is typically too late. It is important that we have a broad variety of these lines available and can test upfront what should be the difference to expect. But timing is a problem to use it really for personalized aspects.
So there's another question. Oh, sorry. Go ahead.
Right. No, Thomas brought up a very good point, and it's really crucial to just state no one testing method is going to give us all the answers we need. There's no one human organ on a chip for lungs or an organelle for the liver. That's not going to give us all the information we need. We need a combination of testing methods, high throughput testing methods for safety, toxicity testing. We need organelle and organ on a chip disease models. We also need to look at more directly human biology, whether it's through imaging studies and pathology results and autopsy results. And so it's going to take a combination of many methods to give us the most accurate picture of whatever the disease mechanism, the human biological mechanism we're trying to understand. And yeah, none of these methods that we're talking about today are perfect by any means, but what's wonderful about them and what's great about them is that we can continually to improve on them in a way that you cannot, as Lindsay said, you cannot do with animals. Ultimately, you still can't change the fact that a mouse is a mouse, even if you insert human genes into that mouse. With these other techniques, though, we can continue, and they are continuing to be refined and improved upon. But what I would say is, it's not going to happen as fast as we need unless we get funding directed into these methods in a way that it is not being funded right now. And it's great to hear about all the different agencies that are committing to funding more and more of these testing methods, but it's still a drop in the bucket compared to how much funding goes towards animal testing.
So there's one question for Thomas in the chat about the mini brains, if they can be connected to other organs.
Absolutely. There's in the meantime commercial platforms, but also a lot of self-built ways of connecting different organs. The mini brains are very much suitable for this because they're not adherent to a matrix, so you can just really move a single ball of cells because they're just visible. You can move it into a chamber. We are working at the moment on settings also with a type of EEG that we have them caged with electrodes to do non-invasive recordings. And you can envisage this all in microfluidics and combined with other organ systems. It is just a matter of who does the work and where's the interest. Do you want the liver combined with the mini brain? We combined it, for example, with blood-brain barriers because that's an obvious one. You can combine it with blood cells as an immune compartment. The limit is the sky.
Thank you. So we have several questions on how we can move away from animal experimentation towards the human biology-based models. And one of our participants is asking, now seeing how different SARS-CoV-2 affects humans versus animals, how can we use the current pandemic situation as an opportunity to stimulate a fast regulatory transition towards non-animal technologies in drug testing and development? And also she's asking how we can increase the acceptance of non-animal technologies amongst researchers. So two big questions. We had several ones in this direction, and I would like to hear all your thoughts.
I can just quickly just say something about the last part of the question or the last question. So what we are finding, and I worked at the Food and Drug Administration for 10 years, and I will say that they are so wedded to using animals because that is what they are familiar with. That is what they are comfortable with. And so for us to really shift the paradigm, we need to help biomedical researchers become more and more comfortable with using these tools and becoming more confident in using these tools. So at CCS, Center for Contemporary Sciences, that's going to be one of our main goals. And we're going to be working primarily at the basic biomedical research level, where there is no regulatory requirement to use animals. They do not have to use animals at basic biomedical research. And so our goals is to get these tools into the hands of the researchers at that phase to increase their confidence, with the idea that will then kind of become more pervasive throughout the biomedical field and will help influence our regulatory agencies at the final stage.
There's a couple of things to do. Yeah, the first thing is, from my point of view, education. Not only the next generation, but also current researchers need to get access to the information, what these methods can do. And this includes definitely the agencies. So we are already working for a while on a certificate program. We have launched courses on Coursera on these new methodologies for free, which have now been engaged 9,000 people in 2 years. Yeah? So really, it's an incredible success. People want to know about these methods. The next step then is to really help that these methods are available. And we should not underestimate that the role of companies in standardizing and disseminating new technologies. They go from lab to lab and try to convince with their beautiful materials, try out this new approach. That's an important factor, which is, I see, very positive. Yeah? It is a force behind change. We also need to instill doubt. We need to spread the information about why these methods are not solving our problems and why we have to change. Why it is unacceptable that we are continuing to use, especially in toxicology, methods which are 60 years and older. We need to make people understand. And the important part, if you're talking to scientists, is do not come from the animal welfare aspect, because they have long made their, can come to a conclusion whether they accept or do not accept these tools. It is very rare that you will shift somebody, that there's a turning point in the scientific life and now I stop using animals. But if you come with the relevance aspects, if you show that you're doing things which are not meaningful, which are not predictive, things which are not solving on an economical scale the problems we have, cannot test enough substances, are too costly in executing them for tens of thousands of chemicals, that's a different story. This is why we're pushing for economical discussions in this, because I know on which side of the ethical side I am. Yeah.
Other thoughts?
And if I can add, yes, that's key, is to develop that business case. That's going to be really key, that economic business case to drive science forward.
Building off of what Thomas said about doubt, we have seen with the COVID vaccines that at least I know for the first one, the Moderna vaccine, it was fast-tracked into human clinical trials without that typical package of animal studies. And you could see doubt in the value of the animal study based on what the head of Moderna said. I forget the exact quote, but it was something like, we don't see a lot of value in this information, and they didn't think that they would learn anything from it that would justify not moving into clinical trials given the pandemic. Now, unfortunately, the same day that the clinical trial started for Moderna, the NIH started doing animal testing for that vaccine as well. So yeah, instilling the doubt, but then getting that to lead to behavior change, I think is going to be quite a challenge.
I just like to make a point about education, actually, and about some efforts in the European Union to try and just raise awareness, I guess, of these the methodologies and to create more resources for them, because I think sometimes maybe maybe some people just don't know really where to look. They don't, okay, so you can look on PubMed, but it's kind of difficult to sort through all of that. So Humane Society International worked with the Joint Research Centre, which is part of the EURL ECVAM, which is the European Centre for the Validation of Alternative Methods. And we did a project looking at, Thomas, you know that very well, don't you? Of course, you know what ECVAM is. You've heard of that. So
my previous job,
yeah. We carried out a project where we tried to, well, we have built a database of non-animal models for respiratory tract disease, and that's one part of this. So there are various other projects ongoing as that, and eventually what this project aims to build is a database of non-animal model resources, which will be accessible for not just people that are interested in research, but also people that have maybe got project applications to review that has an animal model in there, and they can look at this resource and think, actually, there is a valuable alternative non-animal method here, because I think that's what we need as well, is to introduce people, like Aysha said, that are wedded to their animals. Where do they look if they want to look somewhere else? I don't think they really know. So it's about creating these openly accessible databases and training courses, and then trying to push them out to everybody, so that the people that use animals can use it without feeling scared or worried that maybe that's not the kind of environment for them. But you know, this is a resource for everybody. And the other thing that I just wanted to mention ever so quickly was, again, this is an ECVAM project looking at non-animal derived antibodies, and they've created a fantastic document that I can probably put it in the chat, that just really puts the case forward that there is no need to use an antibody for an animal ever again, because the non-animal derived antibodies are better and more sustainable and more reproducible. And actually, how do we get that now out to all the scientists? I think that's a big question for me, is how do we get people to trust us that, you know, we have information to impart that would be useful for them.
Thank you. And can I add to what Lindsay said also? So I'm so glad to hear what you were talking about with the databases. We're going to be doing at CCS something very similar to that, if not the same, in the US. This is one of our first big projects, was to create as much of a comprehensive database as possible of all the different institutions, private companies, governmental centers, academic centers that are creating these new tools. It's a database that will be freely available online. You can learn about what tools are available, what their applications are, who's developing, how to contact them. And so our focus initially is on the US, but we hope to broaden it. But there are so many just within the US itself. And you're absolutely right, Lindsay, there's no like one-stop source, or, you know, 2 or 3-stop source for this information for researchers who may be interested in applying a tool into their work.
Thank you for that. I would also like to point out, you were all almost all involved in that. Our summer school we had in June at Johns Hopkins University, it was virtual. We co-organized that with PCIM. And there's actually a portal where you can see the slides and most of the presentations and so on. And we had international participation, and it was really a great event. And it also showed the desperate need for more education in the field of non-animal approaches. So this is actually the sister event now to the summer school that ECVAM, or JRC, the Joint Research Center of the European Commission, is doing. And yeah, we definitely need to have more educational programs where students can really focus on non-animal approaches, and they don't have to use animals. Because another thing we haven't discussed at all is, of course, many have ethical issues also with the use of animals, because as we know, yeah, there is suffering involved for those animals, and obviously there's something that should safeguard the animals. That's the harm-benefit analysis. In Europe, that is actually something that's required to be done by the researcher and to be assessed by the competent authority before granting a license. And it's just oftentimes the harms outweigh the benefits. And there's one question in the chat saying, why are the economics behind the preference of animal use in laboratories less spoken about? Genetically modified mice and breeding animal-derived products like fetal bovine serum many acknowledge the cost benefit of adopting animal-free innovation, but there seems to be a bigger economic interest in those, not just as ancillary markets. So yeah, the question is, why is, why don't we focus more on the waste of resources, like waste of money? Obviously, we have all those animals harmed without justification, because as we have seen through your presentations, is that mostly it's not good science to use animals to predict human outcomes. So maybe we can go a little bit into the psychology side as well as the economic side, and also how we can do more to promote the non-animal human biology-based approaches.
I think that's a very important one. I stress this, the economical aspects. The most important omics is economics. Yeah? I think we are too much siloed. As scientists, we typically are comfortable to talk about science. We're not comfortable to talk about economy, about policy. And when we first started to go into this topic in 2008 with a small series of articles with Anna Maria Bottini, we found nothing. Nobody had addressed this topic. And quite remarkably, these articles, including the one from 2018, I see this on ResearchGate, where I put all my articles. They are among most read and most downloaded articles I have. Yeah? 5,000-10,000 times for these articles on economical aspects. Obviously, this hits a nerve, but it has not led to a lot of copycat and replications. People shy away from the economical discussion. And one of the biggest lessons of all of this was we are much too much looking into our continental views. A European database, FDA-based oriented database. Sorry, no alternative method will be used by a company which is working globally until the last important economical area has accepted the alternative approach. We have seen this, for example, for pesticides. The one-year DOC study was buried by Europe and by the US. So, we'd say, job done. No, Brazil required it, Korea required it, and Japan required it. And it took us now 10 years to get Korea and Brazil to abandon it, but the fact that Japan is still requiring it means that every company puts their pesticides for 1 year into DOCs, which has been shown to be a useless animal experiment and has been abandoned by all of the others. That's crazy, but this is an economical hurdle, and there's many of these. As I said in my talk, FDA for pharmaceuticals is the to-go point. If you don't convince the FDA, forget it. Yeah? It doesn't matter whether EMA is accepting something in Europe. It is only about the FDA. This is why we should be so grateful that they have endorsed, to an extent never seen before, a testing methodology, which is the microphysiological systems.
And just before the others also answer, there's a question that relates to this whole topic, and it's about what are the power structures that favor animal testing, and who's making the most profit of not advancing to non-animal alternatives?
I think probably the ones making the most money are the contract research organizations who are being paid to do much of the animal testing work now.
And Elizabeth, do you want to say a little bit more on, I mean, you already touched on it in your talk, but do you have to add anything to that? Because it's really, I see that I used to be an inspector, a state inspector in Germany for animal experimentation. And yeah, when I got in touch with the relevant authorities regarding not having certain tests done on animals, they mostly just didn't want to get their hands dirty. I mean, they basically, if they weren't sure, they would rather say, let's do the animal tests. So what are our chances of, like, what is your approach there? Can we train them better, you know, the regulators to be up to date with the new methods, because that seems to be a huge problem. what else can be done to put pressure on the regulatory agencies to be on, you know, at the same state of scientific knowledge, basically?
Well, I do think that training the regulators clearly is something that needs to be done. If you have spent years working on reviewing documents and looking for things in certain ways, and you're not familiar with a new approach, then just, I think it's human nature to be comfortable with what you're used to. So I think there's a lot of value there. But where I see a lot of value for pressure on the agency is coming from Congress. Our Congress has a lot of power to influence what federal agencies do and to help set their agenda via legislation, via funding packages. And so I think that we need to be spending more time with Congress, educating them on the issues, and that will lead to things like funding. I already mentioned in my talk, Congress has directed the FDA to update their regulations to allow for human biology-based approaches and not just the animal data anymore. And so I think there's a big appetite on the Hill for more knowledge and for ideas of how they can work on these issues.
Yep. I mean, this is why we have since 2006 a policy program in the US. Paul Locke is leading this from the beginning, and since 2011 one in Europe. I agree, very often this comes from outside. The agencies are to some extent executive agencies. They're executing and putting into practice a certain law, in Europe even more than in the US. And we have no problem really with the agencies and their leadership typically. They are embracing this. It's very often that they have to be forced to continue executing this to the receiving side. If you see that the EPA only a year ago announced to move out of animal testing by 2035. If you read some of the FDA leadership's pronouncements, they read like animal welfare declarations. Yeah? But if you ask the people on the receiving side who register a drug or so, they are the conservatives who said, better be on the safe side. I don't want to underwrite something which later fails or ends up in court. Yeah.
I did want to say one more thing before we move on about education. And this is something that maybe is a positive that has come in 2020. And that's that when schools were all going online, I think professors and teachers were looking for resources to help teach their courses. And I saw a lot of organizations really step up by making what was otherwise only available to their members free for everyone. And so there is a lot that is available for free online now. I believe the Society of Toxicology did this. I know for a fact the American Society for Cellular and Computational Toxicology did this, and I imagine other groups as well. I can provide links for where people can go to find these free resources. But maybe that's one good thing that we can all be thankful for.
I agree with that, Elizabeth. So sorry. I was just going to say that okay.
Maybe Lindsay,
I just want to say we saw this for our Coursera courses. Yeah, we had typically every week about 30-40 people joining us in each of the classes, evidence-based toxicology and Tox 21. With Corona, it was suddenly 90 to 100. And even now we see about 60 joining every week. Yeah? So there's an enormous demand for online education, and we should take advantage. Yeah? We want to make our Georgetown course on the 3 hours on alternative methods now one of our next projects, and we need to take advantage that people want to learn worldwide. Yeah.
And I think one of the kind of, as I say it, one of the maybe flip sides of that, or another advantage of that, is that we can get more public interest, and maybe we can garner public support for this and public understanding of maybe how drug development works now. I think, you know, it's easy for us to understand how it all goes on because that's all we think about. But for somebody who's just getting a drug out of the cabinet and not even thinking about it, they maybe don't realize that there are other ways to do it. And there are other ways that still allow you to be safe. So I'm not saying that we stop everything right now, but there's a path that we could move towards, and maybe helping the public to understand that would help us get that support that we need. I think the public are incredibly important and powerful in that. And we look at the European Citizens Initiative that was a few years ago that brought up the European directive that regulates animal experimentation, that wanted that reviewed. And that's been incredibly successful. It got its 1 million signatures, and now the directive, you know, it will be addressed, and hopefully we'll be able to make it a bit better. So I think maybe reaching out to the public and trying to educate them as well as our fellow scientists is a really good method for us.
Yeah, that sounds like a good idea. It seems like it is a little bit of a vicious cycle here, because, you know, we have this lack of people working in this field. We have maybe motivated students, but then there's not enough for them, you know, not enough courses, not enough labs they can go to and whatnot. Then we have the problem with the funding. We haven't really, we touched on that a little bit, but maybe we can try to envision how we can shift the funding away from animal experiments, because as we have seen through, one tool is the retrospective reviews, which is a great tool, obviously, like systematic reviews and so on. We have seen that animal experiments are not as predictive and as we hope. And so it is time to move away from that. But there's so much resistance. Obviously, it's understandable if you have built your career in animal experiments and when you've worked in that for decades, it's really hard to change away from that. But the problem is that we need to kind of make this shift away from animal experimentation, and for the young people it's really difficult. So there were several questions around that, like how can we how can we motivate young scientists to get involved in non-animal science, especially since there are not too many opportunities, and how can we shift the funding? What ideas do you have?
I can start with the funding. I think really you have to take a dual approach of working like with the National Institutes of Health, which funds so much of our animal research, and then with Congress, because as I said before, Congress appropriates funds, and Congress can make demands of the agencies for how they spend their money.
And thank you for that. That's really what it's going to take here in the U.S. for shift of governmental funding. And then another avenue is to help bring investors to the private companies, to help inform investors of the potential investment opportunities that are available for them to invest in these new companies, these companies that are creating these tools. In regard to students, what we are doing currently, our organization, is we started a small pilot study to gauge biomedical students' interest in learning about these new tools and gaining their PhDs based on these new tools. We are going to expand that to then become a larger study, and also to look at undergraduate students and high school students whose intentions are to move into this field. What we hope to do with that is, and what we're hoping we'll find, is that there is a great desire on the part of students to learn about these techniques as opposed to learning about animal experimental techniques. And that's something that we can take to academic institutions to work with them to create more programs, academic programs for students. That's just one step. So I think globally what needs to be done is to really get that information from the students, what are their interests, and then take that information to academic institutions. Because I think, and we'll test this out and see how well this works, but I think that's one way to influence the academic institutions, along with trying to find ways to bring funding to create these academic programs that can train students in these new tools.
Any other thoughts on this?
I mean, the science follows bread, yeah, as we say for the arts. Yeah, in the end, scientists will work on where their grants are successful. At the moment, we are writing 1 in 8 grant applications to be successful, sometimes 1 in 15. It's incredibly low, the success rate. So, which means wherever you're successful, this will determine what you're doing in the next few years. This means what are the projects you put your students on. That's creating and shifting the scope of an entire laboratory. So it is extremely important how these decisions are taken. And the funding organizations have an enormous role here. And they often shy away from directing this too much. They leave it to the peer review process. The peer review process is very often dominated by the people who have the time, which are those who already retired. And so they are much longer influencing what science looks like than their actually end of career. So we don't solve these problems one retirement at the time, but really only one gravestone at the time.
Yeah, I think most of you or many of you have seen a paper that was recently published. It actually came out 3 days ago by Don Ingber from the Wyss Institute for Biologically Inspired Engineering at Harvard University. And it has the title, Is it time for reviewer 3 to request human organ chip experiments instead of animal validation studies? That was the question. And yeah, I would like you to comment on that question.
I think the short answer is yes. It definitely is time. And I think, so one of the points that, that Don Ingber was making in that paper was that it's, and this kind of fits with what we're saying about the whole sort of vicious circle that, and what Thomas has said about review is that a paper that goes into a journal that is purely based on in vitro data could very well go out to reviewers who have very little experience of in vitro data and are much more comfortable with in vivo and or whole animal data, and will therefore make a request that that paper, that the authors of that paper go back to the lab and revalidate what they've done in their in vitro model in an animal. And the point there is that that's just backwards. That's absolutely backwards. There's no need to do that, because once again, like Aysha said, you're holding up an animal as a gold standard. This is exactly what we want. And it's really not. It's time that we looked at what's the question that's being asked in that research paper. Is it a human relevant question that's being answered appropriately by those models? And what's the value of that animal data? Really, truly, what is the value of that animal data? If it's not legally required for some sort of a regulatory application, why do we even need to ask for that extra animal data? Surely it's time that we let the in vitro data stand alone, stand by itself, and start to have confidence in it at that level at the very least.
Any final thoughts on that? I think we should wrap it up. We are already over 2 hours. Any final thoughts you have wrapping this webinar up? How should we move forward from here?
I'll just say that I am so pleased with the number of participants we've had today, and it really does speak to the growing interest in this issue. And I actually, like Liz, I do have a lot of hope. I think the tide it's slowly changing, but it is changing. And with so many organizations working on this, both in the US and abroad, I see that within the next 10 years, I do see that tipping point happening. So if nothing else, I hope to leave everyone with a sense of optimism. Yeah, we've talked about all the challenges or many of the challenges that we face, but I hope to also leave people with the sense of optimism that I've been in this area, working in this field for more than 20 years. And for the first time, I really do feel that optimism that the tide is going to change.
Yeah, I can only echo this. I'm 10 years longer in this business, and we have to see the concept of alternative methods is 61 years old. The first 30 years, there was nothing. Then there was a little bit in the 70s and then in the 80s. And then now this is exploding. Yeah, we have, I'm absolutely convinced that we are in logarithmic growth. It will be only a matter of time, and we are on the right side of history. That's the important one.
Yeah, I think it's pretty incredible to Our vice president at PCRM, Christy Sullivan, today is giving a plenary at the Michigan Society of Toxicology meeting. And when I heard that, I just thought that is really something, and that it supports that the tides are changing. When we first were interested in attending SOT, it was hard to be even accepted as a participant at this conference, being people who cared about animal protection as a main goal. And now Christy's giving a plenary. So things are changing, and we really appreciate all the support from the nearly 200 people who have spent 2 hours with us today. Thank you so much.
And yeah, I'd like to thank everyone for being with us as well. I agree that this is, it is a sign of the times that we can attract this many people to these webinars and that we can keep them interested for this long. And I think it's a sign that maybe people want change. And I think if we think of announcements like the EPA reducing their animal testing with a view to phasing it out completely in my lifetime, that's amazing. I didn't think I'd ever see that. And then I think of the support from programs in the National Institutes of Health for the tissue chips and the patient chips and the human chips, and just that kind of backing just makes me feel like there's still a long way to go, which is good because it means we're not all out of work, but we're going to get there. And I think we're going to get there, well, sooner with all of us together. So it's really great to have you all here, and thank you, Kat, for inviting us on.
Thank you so much for sharing your expertise with us today. It was a wonderful session. And I want to announce the next session that Charlotte, Eva, and I are organizing in this series on animals, climate change, and global health. And that is about animals in crises. And that will take place on Wednesday, 11th of November. And we will send a follow-up email to all the participants of today. We will send the recording and we will upload it to our event website. And all the participants, our experts, will share some resources, a list of resources where you can read on further. So thanks everybody for participating today, and have a wonderful rest of your day. Take care. Bye-bye.